CJD is an example of a prion disease and one of the most well-known forms of neurodegeneration. Why are they called prion diseases? It's certainly not after the fairy prion, Titi Wainui, a seabird around the shores of New Zealand. Prion was coined from the term protein infection particle, with PR and ION being derived from protein and infection. All humans and most organisms have the normal prion protein, which does not cause disease. This normal prion protein is encoded by a gene called PRNP on chromosome 20. Its function is unknown. The normal prion protein has a second possible configuration, often described as misfolded, which is both contagious and lethal. In these misfolded forms, prions become resistant to being broken down within the cell. A single misfolded prion protein is sufficient to cause a prion disease because it spreads through what is called conformational influence. A prion will bind to a normal prion protein and cause the latter to also misfold, and so on, resulting in a cascade of infection that can move from cell to cell in a killer domino effect. The misfolded protein can't be broken down and forms amyloid plaques. The normal prion protein has an alpha helical configuration and is protease sensitive. The abnormal prion protein forms a beta pleated sheet and is protease resistant. Histologically, what we see is spongiform change in the grey matter, the presence of many round vacuoles from 1 to 50 micrometers in size in the neuropil. There is also neuronal loss and gliosis. Plaques of amyloid-like material can be seen in the neocortex in new variant CJD. That's the one also called mad cow disease. This is a histologic picture of someone with CJD. You can see the numerous vacuoles present in the neuropil. The red areas are capillaries, the smaller cells are astrocytes and oligodendrocytes, and the larger cells are neurons. This is a close-up version of an amyloid plaque. Notice the similarity to the plaques seen in Alzheimer's disease, but these are associated with vacuoles. The macroscopic appearance in CJD is that of a markedly atrophied brain. Here on the left is a brain with CJD. The ventricles are dilated, the gyri are thinned, and the sulci are widened. A normal brain is on the right. Prion diseases are notable because of the way that they can be transmitted. It can be transmitted by food and instruments contaminated by human brains and other tissues from carriers, and the prion proteins are not inactivated by routine sterilization procedures. They can be destroyed by detergents and enzymes similar to biological washing powders or sodium hydroxide, but contaminated instruments, particularly neurosurgical instruments, must be discarded.